So HCV Research UK is funded by the Medical Research Foundation, and the money is to enable us to establish a national cohort of 10,000 patients from across the UK with a clinical database containing relevant clinical details and laboratory tests from all of those patients, and a biobank. We're recruiting patients from around 40 different centres in England, Scotland and Wales, and we're currently recruiting at a rate of around 400 patients per month, and we're on target to have the whole cohort, all 10,000 patients, enrolled by around September next year. The purpose of HCV Research UK is to act as a resource which researchers can access and use for their own purposes. So we have a system where any researcher anywhere in the world can look us up on our website, hcvresearchuk.org, and can interrogate what sorts of samples we have, what sorts of patients we have, and then make a request that they want whatever clinical data or whatever biological samples for their own research. And we have a tissue and data access committee which will scrutinise all requests for access to the tissue and the data, and the more research groups who use the resource, the better it will be, and we will pass the samples on to researchers to help them in their work. On the basis of HCV Research UK, a multi-centre, multidisciplinary collaborative group put together an application to the Medical Research Council in response to their call for bids on stratified medicine, and that bid was successful and has resulted in a considerable sum of money, around £4 million from the MRC, allocated to do research into why patients do or don't respond to different forms of treatment. And by doing this kind of analysis, which will involve study of the virus, study of the host genome, study of the immune response, and measurement of various biomarkers, we hope to be able to predict which patients will respond best to which forms of treatment. And it's a very exciting time in hepatitis C, with a large number of new drugs appearing on the market, and these are very effective, but they're also very expensive, and some of them have side effects, and it's very important to direct the drugs towards the patients who are most likely to respond. So that MRC stratified medicine bid is called Stop HCV, and it will use the clinical data and the samples within HCV Research UK. And hopefully over a 5-year period we will develop algorithms and clinical management scenarios that will help clinicians to decide how best to treat each patient on an individual basis.
Hi, I'm Sarah. I'm the Biobank Manager for HCV Research UK, and I'm based here at the CVR in Glasgow. We recruit patients and collect blood samples and a lot of clinical data from them from 40 centres around the UK, covering Scotland, England and Wales. Those samples are collected by a team of research nurses, and they're shipped up to us here in Glasgow. They're shipped in these lovely little boxes which are fully compliant with all shipping legislation. For most patients we receive around 10 mils each of clotted for serum and EDTA samples. In a few cases we also get packaging tubes for RNA stabilisation and perhaps PBMCs. These are our standard samples here, so we have approximately 10 mils of clotted sample which will go for serum, and 10 mils of EDTA from which we'll get plasma and DNA. All samples are labelled with a unique barcode, and there's no patient identifiable information on them. Everything is completely anonymised at this stage. These are then processed, centrifuged and spun to give our serum, plasma and buffy coat aliquots from which we get DNA. They are then transported and aliquoted into these little tubes, each which has its own unique barcode again. Each barcode is completely unique, and that's how we identify our samples, which are then stored at -80. we have to be a bit careful with safety if we're working with infectious material. All of our plasticware and sample tubes and anything which has been contaminated with infectious blood is discarded into a pot like this. It is filled with 2% Virkon, which is effective in deactivating HCV. That's then kept for 24 hours and discarded and incinerated. Our samples are all stored at -80 in this freezer and the one next to it to preserve integrity of our samples. That was just a general overview of what happens from our samples from the patient through to the freezer when they're now available for our researcher. Everything about these samples is stored in our database, which can be searched at www.hcvresearchuk.org, and on that website you can also find out a lot more about HCV Research UK.